In this video lecture, we will be discussing on topic starch hydrolysis test. First, we will see the requirement, then we will see the procedure, and then we will see the principle of the starch hydrolysis test. To perform the starch hydrolysis test, we required three things. First, that is a starch agar plate, sterile starch agar plate. Second, that is Lugol's iodine solution, and the third thing, that is our test culture. So what is the requirement to perform the starch hydrolysis test? Sterile starch agar plate, Lugol's iodine solution, and the third, that is our test culture. Now we will see the procedure of the starch hydrolysis test. So for that, what we will do, first of all we will take the starch agar plate. Suppose this is our starch agar plate. Then if we are having the two test culture, then we will make a two partition with the dark bold marker, and then we will mention the test organism name. Suppose here we have a X and here that is the Y test culture. And then what we will do, we will take the test culture with the help of nichrome wire, and then we will streak like a spiral or a zigzag. Okay, like this way. If we have a second test culture, again we will streak like spiral, or you can say zigzag like. Okay, we don't have to streak four flame or direct streaking, just a spiral streaking we have to do. So first of all, we will take the starch agar plate. If we are having the two test culture, then we will make a partition with the help of bold marker, and we will label as one test organism and we will label as a second organism. Then we will just make a spiral streaking of our test culture. Suppose if we are having only one single test culture, so for that, suppose if we are having only a single test culture, if we have been given only one test culture, for that what we will do, we will take a starch agar plate, sterile starch agar plate, and we will streak a spiral streaking in the center of the plate. Then this spiral streaking with the help of test organism we will take and we will make a spiral streaking, and we will incubate this plate at 37 degrees Celsius for 24 hours. We will incubate this plate at 37 degrees Celsius in the incubator for 24 hours. And on the next day we will flood our plate with the Lugol's iodine. We will flood the plate with the Lugol's iodine solution with the help of dropper for 30 seconds. And we will pour the excess iodine. If we are adding some excess amount, if it has been added, that we will pour in the basin. And then we will examine the clear zone around the line of bacterial growth. So what we will observe, suppose this is our plate and here we have streaked our test culture. So surrounding this you will observe the zone of clearance. Here you will observe the zone of clearance, and here you will see the deep blue color. It will give the deep blue color just because of it is a starch agar plate. Starch react with an iodine, it will give the deep blue color. Starch react with iodine, it will give deep blue color. So here only we have streaked. So surrounding the streak, whatever the portion that we have streaked, if organism is having those enzymes which can hydrolyze the starch, then and then only we can see the clear zone. Here is the interpretation, positive test and the negative test. Positive test will shows the clear halo appearance around the colonies. For amylase positive species, if organism is having the amylase enzyme, they can utilize the starch and it will give the test positive. If organism is having no amylase enzyme, that cannot utilize the starch and it will give the test negative. Now we will see the introduction. Starch. What is a starch? Starch is a polysaccharide. It is composed of two polymers. What is a starch? It is a polysaccharides. Okay, glucose is a monosaccharides. Then we have seen the two glucose molecules join. It will form the disaccharides like this way. Starch is a polysaccharides and it is a mixture of two polymer. It is made up of two polymer, that is amylose and amylose pectin. Starch is made up of two polymer, that is amylose and amylose pectin. Amylose, it is a linear polysaccharides, whereas amylopectin, it is a branch chain polysaccharides. Okay, suppose starch, it is a polysaccharide, it is composed of two polymer which is amylose and amylose pectin. Amylose, it is a linear polysaccharides. Okay, whereas amylopectin, it is a branch chain polysaccharides. Why amylose is a linear? Because it has a alpha D glucose linked by a alpha 1,4 glycosidic bond. Whereas amylose pectin, it is a composed of glucose unit. It is a primary linked by a alpha 1,4 glycosidic bond, but with occasionally alpha 1,6 glycosidic bond, which is responsible for the branching. It is a branch because of this bond. It make the amylose pectin branch structure and amylose, it make the linear structure. Okay, so what we have to remember? Starch is the polysaccharide. It is made up of two subunit, amylose and amylose pectin. Okay, amylose, it is a linear polysaccharides. Amylose pectin, it is a branched polysaccharides. Okay, it has a two component, amylose and amylose pectin. Amylose, it is a linear polysaccharides, whereas amylose pectin, it is a branched polysaccharides. Structure of amylose, if we are talking about the amylose part, it is a soluble in a water. Amylose part, it is a soluble in hot water specifically, whereas amylose pectin, due to its branch structure, it is not soluble in the hot water. So due to this, the plate of the starch agar plate, it is a opaque milk white appearance. Okay, the appearance of the starch agar plate, it is an opaque milk white appearance due to the one of the portion, that is amylose pectin, it is not soluble in the hot water, whereas amylose, it is a soluble in the hot water. Amylose pectin, it is a branch structure, due to this it is not soluble in the hot water. Okay. Now we will see the principle. Starch molecules are too large to enter into the bacteria cells. As we told you that starch, it is a polysaccharides. Bacteria cannot utilize the polysaccharides because it is in a complex form. So it should be break down in a simpler form so that microorganisms can utilize those things. So starch molecules are too large to enter into the bacterial cells. So the bacteria that secrete the exoenzyme, the name of the exoenzyme, that is amylase. If bacteria is having the amylase enzyme that can hydrolyze the starch, that can hydrolyze means it can utilize the starch. Okay, and it will break down the starch units such as in the dextrin, maltose and glucose. Suppose how the starch is formed? Glucose, two glucose join together and form the maltose, then dextrin, then starch. So first of all, starch will break down to the dextrin, then it is break down to the maltose and further it will break down to the glucose, and that thing, these molecules are readily transport into the bacteria cells to be used in the metabolism, and it can directly uptake those molecule into the cell. As I have told you that starch, it is a polysaccharide, it is in the form of complex. Bacteria cannot utilize this complex form. So, if organism is having those enzymes to utilize those complex form, so if organism is having the name enzyme, that is amylase, that can utilize the starch, and it will break down the starch into the dextrin, maltose and glucose, and this molecule can easily transport into the bacteria cells. In starch hydrolysis test, also known as amylase test, starch hydrolysis test is also known as an amylase test, because if organism is having the enzyme amylase, then and then only it can utilize the starch. We use the starch agar plate. Here in the starch hydrolysis test, we are using the starch agar plate. We are not using the nutrient agar plate. We are using the starch agar plate. It is made from the nutrient agar plate. In that, starch is added. Okay, that will make the plate opaque milk white appearance. The test organisms is inoculated into the starch agar plate. We stick the test organism in the starch agar plate, spiral sticking we are doing, and then we are incubating this plate at the incubator at 37 degrees Celsius for 24 hours. And on the next day, the Petri plate that we are flood with the iodine solution, specifically the Lugol's iodine solution, we pour the Lugol's iodine solution onto the Petri plate. So, depending upon the concentration of iodine, iodine turn blue, purple or black in presence of the starch. Wherever there is a starch, it is a present, okay, iodine will react and it will give the deep blue color. Because suppose this is our plate, we are sticking only in the center. The remaining places we are not streaking, so already there the starch is going to be present. So in the plate we are flooding with an iodine solution. Wherever there is a starch, it is a present, and we are adding the iodine solution, both together it will form the deep blue color. And wherever the organisms will utilize the starch, there we will see the zone of clearance, because at that place, in that particular place, the starch has been utilized by the microorganism. Okay, wherever we are streaking the plate, if organism is having those enzymes, it will utilize the starch. It will uptake the starch from those plate, and there we will see the zone of clearance. Okay. Bacteria capable of producing amylase and oligo-1,6-glucosidase. These are the enzyme. Amylase and oligo-1,6-glucosidase are grown onto the starch agar plate. They secrete this enzyme. These are the enzyme into the surrounding area. If organism is having this enzyme, amylase enzyme, they will secrete into the surrounding area, and there we will see the clearing around the bacterial growth, indicate that the organism has hydrolyzed the starch. So this is what all about the starch hydrolysis test.